Hey, thank you for joining us today to learn more about Edgewise Therapeutics clinical trials for Becker muscular dystrophy. I'm Jody Wolf. I'm the Chief Program Officer for PPMD. I just want to make a couple of comments as we get started in the webinar. PPMD is working very hard for the Becker community. We've been actively involved and engaged on the steering committee for the Becker Education and Engagement Day, or BEAD, as it's called. This takes place this year on Saturday, September 28th at 6 locations across the country. You can learn more about that. And just 6 weeks ago, we met with a group of over 40 clinicians who are building out best practices for the medical care for people with Becker. So more on that initiative will come this fall. We're continuing those conversations and really trying to hone down on the best care for people with Becker. Today, I'm joined by the team from Edgewise Therapeutics, Dr. Alan Russell, the Chief Scientific Officer and co-founder of Edgewise, Dr. Joanne Donovan, the Chief Medical Officer of Edgewise, and Sarah Tenser, the Associate Director of Patient Advocacy. And the team from Edgewise is going to walk us through their program. We'll have plenty of time for questions after, so please type them in the chat as you think of them, and we'll come back to them at the end of the presentation. So with that being said, I want to welcome the Edgewise team, and Sarah, go ahead and take it away.
Great. Thank you so much, Jody. We are so excited to be here with all of you today and share a little bit about Edgewise and our programs and answer any questions that you may have. I am going to kick us off with just a little bit of overview of who we are as a company and some of our investment in this space. And then I'll be handing it over to Dr. Alan Russell, our Chief Scientific Officer, to talk about our mechanism of action and how we are targeting contraction-induced injury in Becker muscular dystrophy. And then Joanne Donovan, our Chief Medical Officer, will talk a little bit more about our specific trials and what we've found. So Edgewise was founded in 2017, and we are focused on developing novel muscle-targeted therapies for muscular dystrophies such as Becker and Duchenne. We specifically focus on protecting the muscle from injury caused by muscle contraction, and Alan will speak more about how this injury occurs and how we aim to address it. Because we're targeting this contraction-induced muscle injury that happens in everyone, our approach is mutation agnostic. And our goal is that our investigational agent, which is called Sevasymptan or EDG-5506, which you'll see later on, is meant to be taken as a daily oral pill and will be able to be used either alone or in combination with other therapies. We're invested in fully understanding the Becker muscular dystrophy landscape. We understand how serious of a condition this is, that once function begins to decline, it is irreversible. Though we know this happens at different ages for different people, the consistency is that once it begins to decline, it continues to decline. We're committed to listening to the community as we develop this treatment, which I'll speak a little bit more in detail about in the following slides. And finally, we understand that injury caused by muscle contraction, which occurs in daily activity, is a key aspect of the Becker disease process. And we believe that by protecting muscle fibers, we can slow the functional decline that's caused by this injury. We'll spend most of the webinar today speaking about our commitment to conducting and supporting research and drug development for Becker, but I'm going to spend just a few minutes talking about these other areas because we firmly believe that all of these elements, understanding the Becker lived experience and supporting efforts to provide education and community building, are all interconnected and essential to fully supporting this community. So in this slide, you'll see some of our efforts that we've conducted to deepen our understanding of the Becker experience. We've had focus groups, advisory boards. We have a standing patient council that's been active for over 2 years now, I think. We've had in-depth one-on-one interviews, and then we also recently completed a quantitative survey of over 50 individuals living with Becker across the United States. Through these efforts, we've seen the extent to which living with Becker is a heterogeneous experience. Aspects of diagnosis and disease management can be very unique to the individual. We've seen how much Becker can impact day-to-day quality of life and how this isn't always quantifiable with measurable clinical outcomes. Again, it's a very nuanced disease. We've heard that cardiac health and decreased mobility are two of the most concerning aspects of the disease. And we've also had many conversations about how all of these things together can impact mental health. And we recognize how important it is to care for that component of the condition as well. And then on this slide, we have outlined some of the efforts that we've made to support education and connection in the Becker community. This first up here is the beckermusculardystrophy.com website that's accessible either by clicking that in. We also have a QR code here, which might be a little small on your screen, but we can make sure is available to everyone. And this has education about disease process, lists of resources, other things that might be helpful as you're navigating the disease. We've been working to put together an educational video about disease process in both Becker and Duchenne, which we'll actually get to show you guys a little bit later. Really excited about that. And then we've also supported different flyers and resources, just providing different education about disease process, clinical trials, other types of resources. The screenshot we have here is from one of the MDA Quest magazines. And then finally, which Jody has already mentioned, and we'll talk about again later because we want to make sure that everyone walks away knowing about this wonderful opportunity, is that we, along with PPMD, are members of the steering committee for Becker Education and Engagement Day. It'll be happening, as Jody mentioned, in 6 sites across the country. We'll be in Seattle, Missouri, Dallas, California, Pittsburgh, and Florida this year for a full day of Becker-specific content, opportunities to connect with experts in the field and to connect with other people living with Becker or caring for somebody with Becker to help build up that personal network. So with that, I will hand it over to Alan to talk a little bit more about our novel approach to protecting muscle in Becker muscular dystrophy.
Thank you so much, Sarah, and welcome to the webinar. Now, this webinar is specifically geared towards Becker muscular dystrophy and the Grand Canyon studies, but we understand a bunch of you are going to be tuning in from the Duchenne side too, and we'll make every effort to kind of loop in some extra information around that, just to give you some additional background. We will have additional webinars, of course, specific to Duchenne and the Lynx Fox type studies. This is our forward-looking statement. We're a public company, and the goal of all of this small writing is just to point out that sevasemten is a compound that's in development but hasn't been approved anywhere. So what we say is essentially speculation on what we hope will be the outcome in clinical trials. And we have a lot of trials going on. As I mentioned, this is specific to Becker. I'll run you through these, but then I'll mention the Lynx ones at the end. So we have the big study going, Grand Canyon. So that's the ongoing recruiting study in Becker muscular dystrophy for people 18 plus. There's the clinical trials number there. This is the one we're really leaning hard into. It's a large study, very large for a Becker muscular dystrophy study, the largest that's ever been run. And it's absolutely essential for us to get people into that study to see whether the compound works or not. So we're doing everything in our power to get people into that study as quickly as possible so that we can get the results and then get those results out to you guys and to the FDA so that we can see if the thing can get approved or not. MESA, that's the extension study. So if you've been in any Edgewise studies for Becker muscular dystrophy, you get transferred onto that open label study. So you keep getting access to the compound and we see what the compound does long term. The active not recruiting, that's the data that we will be revealing at the end of the year. So this is kind of the prelude to Grand Canyon. It's a medium-sized study, but saying medium-sized, it's still the largest study that's been run in Becker, looking at function over a 1-year period in Becker muscular dystrophy. So we're gathering that data right now, and we'll be releasing that data towards the end of the year. And then ARCH is the open label study in Becker muscular dystrophy that Joanne will be telling you about. And we now have 2-year data in that study to give you a better idea what the compound does in the background of Becker muscular dystrophy. And as I mentioned before, we have the Duchenne studies going. This will be the subject of a separate webinar, but we have, of course, the Lynx study and the Fox study ongoing, looking at the effect of sevasemten either in the background of steroid therapy or in the background of steroid and gene therapy. therapy. That sounds right. My job as a scientist is to tell you how sevasemten works, and then I'll hand it over to Joanne, and she'll tell you how it fares in the clinic. So this is some key observations for the messaging around how the compound works, and then I'll show you the new video as Sarah suggested, and then we'll bit more video, and then I'll be done. So some key observations on the top there. Some skeletal muscle fibers are more susceptible to muscle damage when they don't have dystrophin. We often talk about fast and slow muscle fibers, and this is really key to the understanding of how sevasemten works. Scientific reports suggest quite clearly at this point that fast muscle fibers, they make up about half of your muscles, are much more susceptible to injury than the slow ones. And the fast muscle fibers are what sevasemten targets. Okay, so we've made that compound, sevasemten. In previous lives, it was called EDG-5506, is designed to protect those susceptible muscle fibers, regardless of the mutational background. And it's going to do that by changing how the muscle contracts rather than what kind of genes it expresses. Okay, in diseased animal models, mostly Duchenne models, sevasemten protects susceptible muscle fibers from injury, and that causes long-term beneficial effects. We've seen effects on fibrosis, we've seen effects on kind of strength and activity in dogs, and I actually saw Pete Niem was on the call, who did our dog studies. Thank you very much, Pete. And then in initial clinical trials, and these are the ones that Joanne will tell you about, sevasemten quite reliably decreases leaking muscle biomarkers, and we'll tell you about that, and stabilizes muscle function in the background of Becker muscular dystrophy. Next slide. Okay, here's where we get to the video. This video is really an educational video to try and fill in a gap around the role of dystrophin and why it's important to protecting your muscles when they contract. So it's not really around sevasemten, it's really just educating people about the role of dystrophin and muscle injury with contraction. So roll it away.
How muscles work and why they sometimes don't. Our muscles do a lot of work for us. We all know they make movement possible. Every muscle is made of thousands of long strands that contract, creating movement in the body. These contractions put a strain on the system, leading to little tears in the muscle fibers. This is called contraction-induced injury, and it happens to everybody. It's why people can feel sore after exercising. Protecting muscles from too much injury is where certain proteins, including dystrophin, have a vital role to play. Ah, yes, there he is. Look at him go. These proteins connect our muscle fibers together so that strain on muscles is spread evenly when the muscle contracts, similar to threads in a rope working together to strengthen and stabilize the muscle. When muscles are working properly, tearing is minimal and repair is quick. Soon the damaged muscle is back online, doing what it needs to do. Sometimes key parts of the muscle are not working, so the muscle is susceptible to damage from muscle contraction. We see this in muscular dystrophy, a group of diseases that includes Becker and Duchenne muscular dystrophy. In these diseases, dystrophin is only partially functioning, as seen in people living with Becker, oh yes, or totally absent, as seen in people living with Duchenne. That means that when muscles contract, force from the contraction isn't spread out over the muscle, which can damage the muscle fibers. In contrast, in fully functioning muscles, muscle contraction doesn't cause excessive injury. But in people with Becker and Duchenne, damage can be more frequent and severe when their muscles are contracting. The muscle membrane can become leaky and let unwanted visitors like calcium inside the muscle cell, causing a complete fiber breakdown. Over time, the muscle gets inflamed, and instead of being able to repair itself, scar tissue and fat are laid down. When this happens, we often see muscles swelling at first from the stress and then shrinking as they become less effective and are used less. This muscle wasting is also called atrophy. Damaged muscle fibers don't do their job and are weak, which puts more strain on the remaining fibers whenever they contract, resulting in a cycle of continued muscle damage. Over time, the impact of muscle damage caused by contraction in Becker and Duchenne causes loss of muscle and therefore loss of strength and function. Moving around becomes harder. Standing up and walking become a challenge or sometimes impossible, and many people need a mobility device to get around. Although there's no cure for Becker or Duchenne today, medical researchers are looking for treatments with urgency and focus.
Protecting muscle is a key goal in muscular dystrophy research, and there are many ideas being explored to protect muscle from injury caused by muscle contraction and to preserve function, to improve the lives of those with Becker and Duchenne muscular dystrophy. In the meantime, the progression of Becker and Duchenne can be best managed through working with your care team to identify options for protecting and preserving muscles. Your doctor and care team can help you monitor your disease or that of your child and build the most effective plan to manage it.
All right, there you go. New video. That guy is not a relative of mine, by the way. I have no idea who he is. But hopefully the goal here is to really help you understand what dystrophin is doing. It's often described as a shock absorber, and that's just a couple of words. We're hoping to put that more into a visual context so you get an idea of what it's doing. And the real goal of sevasemten, which isn't in that video, but is the topic of today, is to curtail that contraction-induced injury, right? To relieve the stress on these muscles that now don't have these connections, and hopefully change how they contract so that they contract and they don't injure themselves. Okay, next slide. Here it is really in motion. So these are dystrophic muscles. So these are from Duchenne muscles. The left-hand side muscle has not been treated. It's a very small muscle. You can see right through it. This is from the mouse. And when that muscle is contracting, much the same as in the video, the stress that now builds up along those fibers causes the stress and breakdown that you see like real time here. Okay, it's a sped up video. And you can see these clumps of muscle fibers kind of moving together. And that's really as a result of the stress causing the breakdown of the muscle fibers. And then they kind of shrinking in on themselves in a kind of collapsing type of maneuver. And you can imagine if that happens to any extent, that causes considerable problems to muscle health. On the right-hand side, what we've done is treated the muscle with a small concentration of sevasemten, 5506. Now that's changed how that muscle is contracting. It's targeting those fast muscle fibers, and it's slightly decreasing how those muscle fibers contract, the speed and extent of those contractions. And that tunes the muscle to a place that it can contract without injuring itself. Now that tuning is essentially what we're doing in our clinical studies right now. You try different doses, and what you're hoping to achieve is a tuning of the muscle so that it doesn't injure itself anymore, but a tuning that doesn't go too far such that the muscle isn't functional anymore, right? We want to maintain function, but we want to protect the muscle. So that's the goal of sevasemten. And you'll see Joanne referring to this when we do our kind of dose ranging type studies that we've done in both Duchenne and Becker muscular dystrophy. At this point, I'm going to hand it over to Joanne, and she'll tell you about the clinical studies and the kind of results that we're getting and you should expect to see in the future.
Great. Thanks, Alan, and everybody. So what happens when there is contraction-induced damage? And you saw in the video that the membrane, the outer surface of the muscle fiber, is damaged. And what that does, it allows proteins to leak out of the muscle. This is a protein like creatine kinase, CK, that we know is often useful in making the initial diagnosis of muscular dystrophy. It can be other proteins that are measured in the clinic. It can be proteins that are called biomarkers of muscle damage that we may only measure in research studies. But the whole, looking at all of these gives us a very good idea about whether or not there is ongoing damage to the muscle. And if we can prevent damage to the muscle early on, then we anticipate that that's going to translate into protecting the muscle and preserving function long term. And importantly, we can measure these in the bloodstream. So we don't have to do a muscle biopsy to look at changes in the muscle. What we look at is a blood draw, which is much less invasive. So that's one of the things that we do in the clinical trial. How do we measure function in the clinical trial? And that is complicated. What we are using, and one of the things that we are using is called the North Star Ambulatory Assessment. And that looks at 17 different activities that measure function in those that are able to walk. And each one of these is scored by a physical therapist as to whether the activity can be done without compensation, without, for example, like leaning on a piece of furniture to get up or something like that, or can it be done at all? And the maximum score is 34. It's been used by practitioners. It's been used in clinical trials, importantly, to measure how motor function happens over time. So on the next slide, these are the 17 different activities, ranging from the hardest, hop on left leg, to the easiest, standing. And losing each of these activities, which does happen over time, has real world implications for people with Becker. And so things even like standing on heels, if you are stretching, can you walk on uneven ground? Is that giving you trouble getting out of a chair? We look at things like climbing a single step. That's analogous to going up a curb, for example, or going down a curb. Can you do that without holding onto a banister or something like that? Can you get up from a chair? It's important in everyday activities, in toileting, and of course in walking and measuring that. So it measures a whole range of activities over the spectrum. And importantly, we have this information over time on the next slide. So it has been looked at in natural history studies, and the maximum is 34. People tend to lose the ability to walk when it's down around less than 5. One of the other measures that we look at is the amount of fat in muscle with a muscle MRI. And it's not that the muscle can't work, it's that there is actually loss of the muscle tissue over time. You saw that. We tried to express that in the video. But it's the loss of muscle that causes people to lose function. So of course, once that muscle is gone, it doesn't come back. How can we tell you what's going to happen? Can I actually, if you can go back one thing, sorry. How can we tell you if that's going to happen? And it isn't just age. Some people start to lose function in their late teens. Some people lose function in their 30s or even later. But once it starts to happen, that is a predictor that it will continue to happen. People don't lose some function and then, well, I'll just stay here for 10 years. It happens that there is more replacement of muscle with fat and fibrosis. And because there are natural history studies that look at people without any drug intervention, that helps us plan clinical studies because these kind of natural history studies are done. And it shows us that the North Star in people who have started to lose function goes down about 1.2 points per year. And that may not sound like, but over 10 years, that's a lot of functions that can be lost or need to have compensation for. So in the next slide, these are the studies that we are ongoing. The one I'm going to talk about the most is the ARCH study. We have the 2-year data that. The Canyon study, preliminary study, then we moved into the Grand Canyon, the pivotal study. And what pivotal means is the same thing as a phase 3 study, that it is a study that is designed to look at efficacy, to look at how the drug works in people, and be able to go to regulatory authorities and say, here's the information that you need to potentially approve the drug. So that's what we're trying to accomplish with the Grand Canyon study right now. It's important after people are in these studies to be able to continue of the drug. It's important to them to have the opportunity if they feel the drug is working for them. And it's also important for us to be able to get more information on how the drug works long term. So the people who have been in the study, the people who were in the ARCH study, and even people in our phase 1 are in an open label extension, meaning that they know they get the drug and they are followed relatively infrequently, like every 6 months, to look at the long-term effects on efficacy in the drug, how the drug works on function, as well as safety, and get that important information for the regulators. We've also done a study to look at sevasemten in some other muscular dystrophies, limb-girdle muscular dystrophy, and also in a muscle disease that is not a muscular dystrophy, McArdle. And that is more of a proof of concept study to look at contraction-induced injury with severe exercise and show that the biomarkers of muscle function decrease with that. So you'll be hearing more about that later this year. So in the next slide, that first study, the ARCH study, has given us a lot of information. This was the first study where patients were out in the real world. We looked at primarily, this was to look at safety and tolerability. It's the first time people are being treated with the drug. These are men aged 18 to 55 who had a dystrophin mutation and had Becker phenotype. In other words, they were ambulatory in that age range. They weren't on corticosteroids. Those are not used very much in adults with Becker. And they could walk as far as a 100-meter timed test. We enrolled 12 patients, including some of the patients that were in our phase 1 study, which was an inpatient study. And those guys really got a gold star for doing that inpatient for 2 weeks. We started them on a baby dose, 10 milligrams, maybe not a baby dose, but a smaller dose. And then we went up. And what this allows us to do is to look at safety, to look at how much drug is absorbed, and to look at the effect on function, on safety. And so this is part of drug development. What's the right dose? So we started at 10, we moved to 15, we moved to 20. And what I'll show you is that what we found was the evidence pointed to 10 milligrams being the right dose. As Alan said, we are looking for the sweet spot of what the best dose was. And it turned out to be actually a lower dose, 10 milligrams. So this took time. We needed to look at people for several months at each dose to be able to understand this. And that gave us, importantly, that gave us the information to design the pivotal study. So we didn't just look at safety. We looked at functional measures like North Star, and we found out important information. We found out, even with the first study, that the drug was well tolerated. I should stress also it's given as an oral tablet once a day. And in that, before the ARCH study, we did muscle biopsies, and it showed that the muscle is absorbing the drug. In fact, it's 100 times more concentrated in muscle than it is in the bloodstream. So that tells us it's getting to where it needs to be. We've done muscle biopsies. That's not something that we including in studies after this, but it's important to understand how the drug is working. So in the next slide, this shows the biomarkers. So the damage to the muscle. What are the proteins that leak out? Creatine kinase. The second one is fast skeletal muscle troponin. It is a protein that is only in those fast muscle fibers. Alan told you there's two kinds of muscle fibers, fast and slow. The fast ones are the ones that get damaged in Becker and Duchenne muscular dystrophy. And so that's an important signal to us that that the drug is doing what it's supposed to do. Myoglobin, same thing. That is a target that is damaged in muscle injury. So what we saw was all three of these went down, and they went down even with the lowest dose to pretty much the maximum amount that they went down. So what that told us over time was that the dose of 10 milligrams hits that sweet spot. So by going up in dose, it allowed us to figure out what the right dose was. And then we came back down again to the 10 milligram dose. And actually that's the dose that we have in the Grand Canyon study. So the next slide, we also looked at function. We looked at the North Star Ambulatory Assessment, and we looked at how it changed over the 2-year period. Now, these men were men in their 30s typically. They would have been expected based on where they were starting out in North Star to go down by about 2.5 points over that 2-year period. And what we saw was that they were stable over that period of time. Which is good. They're not losing function. We don't want people to continue to lose function, and that's very encouraging. And so having that information and having that natural history study, which patients with Becker have been contributing to over the years in other countries, there's a natural history study going on at University of Florida now. That gives us very important information to be able to design clinical trials. So this allows us to help design this pivotal trial that's next. On the next slide, so what did we learn from the ARCH study? The safety was very good. The drug was well tolerated at all of the doses. So we went to higher doses and still saw safety was good, and then went back down again. We saw that they were rapid and sustained. They kept there over 2 years, and statistically significant decreases in multiple biomarkers of muscle function. And we saw that function was stabilized. And in some of the things that Sarah talked about, what we hear from patients is that it's important to maintain what you have, to not lose more. And we identified the dose. So we had all of the ingredients for the design of the Grand Canyon study. So that's, it was also interesting we did something called exit interviews, which talk with patients to understand what they thought about the trial, their experiences in the trial, and what they expected to do. So this is really what this is more what we're showing you is more what they thought about going into the study, because that's important as people are thinking about going into studies. And I guess that's not surprising for an early stage study like this, that they were thinking about altruistic reasons as well, because I think people's expectations are modest. But the idea of being able to help and potentially other relatives, a lot of them actually had other family members affected by Becker. We focused on asking them questions about the North Star scores and how the items in the North Star could be important in their daily life. And that was something that we got a lot of information about in individual measures. The vast majority of those measures that are looked at in the North Star are relevant, people felt, to their daily life. So on the next slide, excuse me. So the Grand Canyon study, since it was started out as a phase 2, it's still caused a phase 2, but it's a pivotal study, and it's 18 months long. So to look at the changes in function over that period of time. We are enrolling men aged 18 to 50 who have a genetic diagnosis of Becker and who can walk and meet certain functional criteria to show that they are in that range that is starting to decline. You can use assistive devices like a cane or orthotics to complete those. And we are looking to enroll 120 males. Now, why do we pick those kind of entry criteria? Because I did see a couple of questions pop up. And when we're designing a clinical trial and we want to design it with an understanding of what's going to happen to what we would expect to see in those people over time. So then we can actually see whether or not there's a change in people who get the drug. So that's why we are somewhat limited in looking at the people who have been in the natural history studies. For example, in kids under 18, we've actually enrolled almost 30 adolescents in our earlier studies, and that will give us important information on biomarkers. But it turns out that function doesn't very much change in that age period. So what you want to be able to show is that something changes over time. And if it doesn't change, then you can't really show a difference with a drug. So that's why we didn't include, for example, adolescents in that, in the study. Then that's why we are looking at men who are ambulatory, because there's much better information on how things change over time in that group of individuals. On the next slide, this is what the study looks like overall. First, there's a screening period where you are, there are certain tests to make sure that it's safe for you to participate, that it is, that you meet the criteria for entry into the study, like review your genetic diagnosis. And then over the course, including that one, over the course of the 1.5 years of treatment, there's 7 site visits, 18-month treatment in all. And at those, so they are spaced out at a month, 3 months, then it spaces out to 6 months, then 12 and 18. There, at those visits, we do blood tests, physician examines you, we do functional assessments. And at the beginning and the end of the trial, so only every 18 months, there's an MRI. There isn't muscle biopsy. You take the drug home, take it every day. And we do some other functional tests as well. And as well as ask you questions about how the drug, how you feel differently during the trial, how you see things in your daily life going on. So it's mainly about function. We have, on the next slide, we have tried to include sites across the country. People in this age range are busy. They've got families, they've got jobs. So we want to minimize the travel requirements. So there are quite a few sites in the US, although I realize there are states that don't necessarily have, but there's a lot of sites. We're also doing the study in Europe and Australia, New Zealand, and we are supporting people in terms of travel and reimbursing for travel. There's a little bit more on that in the next slide, on the next one. Maybe I'm sorry, I thought that was in there, but we are reimbursing for travel. What we do is we make sure that we pay in advance for anything we can. We reimburse for things like childcare that are, so the idea is that people are not out of pocket for participating in a trial. And there is a stipend for people's time as well, because it takes time, you guys are working. So to summarize, sevasemten, it's taken orally. It's intended to preserve and to protect the muscles and preserve function in individuals living with Becker and with Duchenne. The goal is to prevent damage to muscle by protecting the most susceptible muscle fibers, which is a key aspect of the disease process. And we see this potentially used alone or in combination with other therapeutic approaches, because this muscle damage is ongoing, whether people are on steroids, and things like CK are still elevated despite other treatments for example, in Duchenne. So, lastly, we are, as Sarah said, committed to understand the burden of trial and to try to make these as patient-centric as possible, providing support services, prepaid expenses, and to try to reduce that travel burden, because when people have limited mobility in particular, that's even harder to be able to travel to sites. So to summarize, this is as you well know, it's a serious disease, and whether declining function begins at different ages, once it begins, individuals are really on a path to irreversibly losing muscle and therefore function. There's multiple challenges, and we need to listen and to understand that we are addressing those challenges and can correlate what we're measuring with what people are challenged with in daily life. And it's this contraction-induced injury that really happens with daily activities of daily living that is something that we can address and potentially protect muscle fibers to slow and stabilize individuals from decline. So with that, I'm actually going to ask Sarah to tell us more about Becker Education and Engagement Day. She has been very much involved in that.
Yes, wonderful. So there's a QR code on the screen, as well as our website, beckereducationandengagement.com. You'll see here all of the different wonderful groups and institutions that have been involved with this, including PPMD, to bring together speakers around cardiac health, mental wellness, physical therapy and exercise, all different topics that we've heard from the community, both from this event last year. Last year was the inaugural year, and then also from different engagements throughout the last 12 months to help inform that agenda. It's free to attend. There will be meals provided, and the states where it will be hosted are listed here. So we're happy to answer any questions about what that will be, but it'll be a really amazing opportunity. We have, I think, over 170 people have already registered for the event coming up at the end of September. So be sure to grab your spot if you're interested, and reach out if you have any other questions. We can leave this up for a minute, but I think, Jody, if you wanted to start kind of going through the questions, and I'm happy to navigate back to any other slides that might be helpful to have on as we're answering questions.
Great. I just want to thank the Edgewise team, Joanne, Sarah, Alan, so much for presenting this information in such a way that is able to be understood and digestible. We have a very active chat going on right now of Q&A. So I have been tracking that, and we're going to get to as many questions as we can. And I just want to encourage the rest of the audience, if you have more questions, to go ahead and type them into the chat. So let me start with a question we had about the mechanism of action of the drug. First of all, the video is great. And
yes, I think that's a me question, Jody. There's lots of questions about mechanism and that.
So the question specifically was around, is it a relaxant? Does it relax the muscles? Does it put the muscles in a certain kind of hypofunctional state? If you could address that, Alan, thank you.
So the mechanism of action, I'm going to get technical, so warn yourself. The mechanism of action of sevasemten, it is a type 2 fast muscle myosin inhibitor. Okay, so myosin is the protein in muscle that modulates contraction. It's an enzyme, and by consuming energy, it actually generates force in the muscle. Okay, when I think of a muscle relaxant, that's kind of things that you take to help your muscle kind of calm down and relax. This is not what it's doing. This is actually kind of a speed limiter for muscle. So the more compound that you use, right? So as we go up in doses, the more you kind of put a limit on the top speed of your engine, right? If you took a massive dose, then your top speed would be very slow and your muscles would be very slow. Okay, that's not the goal. The goal is just to take the edge off, to take a little bit off the top. So it's not relaxing the muscle, it's actually like limiting the speed. Okay, does it put it in a hypofunctional state? Well, that's not the goal. The goal is to choose a dose that stops the stress but doesn't slow down the muscle to a point where you would notice it, right? Because that's going to make things very difficult for everybody if we go up to a high dose like that.
So the so if we could go back and talk a little bit about the biomarker data, and maybe go back to that biomarker slide. We had a number of questions about that, about how did you choose the 10 milligrams? A couple of people made comments that it looked like the 15 milligrams really dropped the biomarkers and you would assume that would be good. So why go to the 10? And then I guess at the end, it maybe looks like the 10, it's kind of inching back up in a couple of cases for the troponin I and the CK. So just walk us through this a little bit more.
So as Alan said, kind of more is not more. And what we want to do is we want to protect the muscle. Now, is it any better to protect it at 85% versus 80%? Probably not. And so when you look at, these are the error bars, these plus minus, and that's what are used to show kind of, well, it isn't a point, it's somewhere in there. And these are not different at 10 milligrams, and I realize which is at the end of that lighter period versus the end of 20, for example, for CK. It's a tiny bit more for fast skeletal muscle troponin, but that's a marker that's a little bit more bouncy. You see the error bars are bigger there. And the same for myoglobin. It's really not any different at 10 milligrams than it is at 20 milligrams. So that's how we pick the dose, basically, to get essentially near maximal decrease in the biomarkers. And we have a lot more biomarkers than just this, I will say. There's a whole panel that we look at, and it gives us the same answer. What we also see is that over time, we see these biomarkers go down. We also see as we go over time that the inflammatory biomarkers tend to go down as well. And they don't change with different doses. It's more of a time effect. So that's where we kind of came out with the dose here. That is the dose that gives us the near maximal. And of course, when you talk to regulators, you want to use the lowest dose that works, basically. And we're not getting any more out of the higher doses. And you don't really see that also in the functional measures either. You don't see that you get more protection. Now, related to that is, do you expect it to just protect the muscle, or could it improve the muscle? And when you think about how you, in daily life, if you really push yourself, if you climb a mountain, your muscles are sore the next day, and they are leaking some of these biomarkers here as well. The other thing that happens the next day is if they're sore and they're damaged, you're not as strong the next day because you're sore. You can't push yourself as hard. So we know that some of the people in the study actually did improve over the 2-year period. And that, is that because their muscles aren't in this constant state of ongoing damage to the muscle? One other thing I just wanted to add to what Alan said about, I know I'm keeping going on here,
No, you're
about
fine.
if you did go to a high dose, you're not injuring the muscle, you're inhibiting the muscle. And this is a small molecule. So if you take it away, then that effect goes away as well.
Okay, thank you,
It's
Joyce. I
different.
think you did a great job explaining the dose, and we had multiple questions about that as well. Another question was, have you seen in your studies a correlation between someone's mutation and how they respond to the drug? And if you have it, how are you examining this moving forward?
Yeah, so we do look at the mutations. We collect that information. And in some of the natural history studies, that also records that information. There are certain mutations that tend to not have a very severe disease. Turns out we have none of those in the group that was enrolled in the ARCH study. They're very much like the mutations that are in the natural history studies. A lot of the mutations just in general are around 45, 47 to a few exons beyond that in frame. And so those are the mutations that are common in this study.
Do you have any out of frame mutations with Becker phenotypes in the
They
study?
do, they're not in the ARCH study. About 10% of people who have a Becker phenotype, in other words, are walking past 18, have an out of frame mutation. It may be, and I will get in trouble, it's a mutation that there is some ability to compensate for in the splicing. But as long as they have a mutation in the DMD gene and are able to walk, then they are allowed in the study. And again, about 10% have an out of frame mutation. There's a lot of mutations that could be either Becker or Duchenne, depending on other genes.
Great. And speaking of mutations, we had a number of questions about manifesting carriers and women. How are you thinking about that? Because you're studying this drug in both Duchenne and Becker, so a broad dystrophinopathy population. How are you thinking about the women?
Mm-hmm. And I saw that question. You're right. Women who are manifesting carriers have less dystrophin, and so why not include women? There are natural history studies that are ongoing. I know that Nationwide is conducting, and PPMD has supported that to understand what is the course of disease in women. But without that information, it's hard to include in a clinical trial because we don't know kind of what would be predicted to change over time.
Okay. So you're saying we need to establish the natural history of
That's a really important study and
Okay.
I would encourage you to be involved in that.
But it is
Theoretically, though, go
it's an
ahead,
active
Alan.
area. It's an active area of interest for us. We're going to do some biomarker work in carriers. We need to kind of set the table. To be honest, in order to do a trial, you kind of need to understand the problem.
Okay, great. So you are thinking about the women. And theoretically, scientifically, is there any reason this wouldn't be an option?
Not at all.
Yeah, and I think the biomarkers help because if we see if we are, when we're able to show a change in function, and that we see a biomarker change and a change in function, then that allows you to say, okay, we've seen protection of the muscle with the biomarkers that translates into function. And then for smaller groups of patients, like boys where we don't have natural history, and the younger boys under 12, if we look at biomarkers then, and we look at safety, then that is potentially enough for regulators to then say, yes, that should be okay if you're seeing the changes in biomarkers that in other groups have predicted improvements in function.
Great.
And there was just a specific question about, can you just reiterate who was doing that natural history study?
It's at nationwide.
Yeah, nationwide.
Nationwide?
And we can send out additional information about that.
Mm-hmm.
Dr. Ma, isn't it? Okay.
Can you talk a little bit about the cardiac function? That's such a huge
Yeah, there's a couple of
issue.
questions in there. The compound is designed to be exquisitely selective to just that fast myosin. Okay, there's different types of myosin to different types of muscle. So there's a different one in slow muscle fibers, and there's a different one in cardiac muscle as well. And the compound does not target those two forms. So in early safety studies, we took a close look at cardiac function, and it wasn't altered in sevasemten treated preclinical models or people, right? Now, that doesn't mean that the compound couldn't have a beneficial effect on the heart in, for instance, Duchenne muscular dystrophy or Becker muscular dystrophy, where long-term kind of muscle dystrophy gets your heart too, right? We've seen in preclinical studies, specifically in the DBA2J mouse, where we treated them, the heart actually got better. There was less fibrosis there. Now, presumably that wasn't a direct effect on the heart. It was indirect from the skeletal muscle, but that's something we're keeping an eye on.
Are you tracking it in your trials?
Yeah, we are doing echoes.
Echoes, so,
Yeah.
but not MRI.
Yeah, we've talked with actually many of the MRI experts and to come to, yes, that, and again, it's a balance between burden and getting the information. So.
And are you doing muscle MRI in your trials?
And we're doing muscle MRI. Yes, we are.
Can you talk a little bit about the side effects?
Mm-hmm. The most common side effects are dizziness and some sleepiness. We've been giving the drug at night, and that seems to minimize that. Interestingly, it tends to happen at the beginning of dosing, and then tends to disappear, actually. So even when the least amount of drug is in the body, and we think it has to do with, it's something that the body gets used to, and it may have to do with how the body senses where joints are with limbs, and that some of the muscle fibers are involved in that.
Great. And we've had a number of questions asking if you could just go through your data readouts. What should the community be expecting and when for both Duchenne and Becker? Walk us through that, please.
I know there's questions on Duchenne as well. So you saw that it took a while to figure out the dose in Becker, and we're doing the same thing in Duchenne. What we do is we have, that is a 3-month placebo-controlled study. We're going up very gradually in dose and looking at safety along the way. So that we have to look at one group of 3 months and go up. And we have, and some of the boys have only been on for a few months at this point because we are watching very closely. And so we're anticipating that we are going to provide some more data the end of this year on the LINK study. I know that it feels like a long time. We are trying to make sure that we find the right dose, and that we saw in Becker that involved going up and coming back down again. And that's where we are also, in trying to understand the dose with Duchenne as well. So that's the end of this year. The end of this year, we'll also have the Canyon study, the first part of the Grand Canyon, which is 40 adults and 29 adolescents, 12 to 17. And that end of this year, we'll have a readout on that and be announcing that.
So you'll have
those are the two.
69 patients of data at the end of the year?
Yeah.
For Canyon and Becker?
Yes.
Great.
By far and away the biggest study that's ever been run in Becker
Yeah.
muscular dystrophy.
Yeah.
Yeah, that's fantastic. We'll be looking forward to seeing those
And,
results.
we did have some questions about the size of the Grand Canyon study. Do you
Yes,
just want
that's
to talk about
120 patients. And is that enough? That is what we calculate the size of the study based on how much variability there is in doing the measure, as well as how much change there is in the measure. And if a measure is more variable, then you need a lot more patients. It turns out that, not surprisingly, adults are better at following instructions than 4-year-old boys are in clinical trials. You may be surprised at that, and so we actually, given how variable the adults are, pretty not variable when we repeat the measures. And so it turns out that we are very well powered at over 90% with 120 patients. So.
Right. I see that we're coming up on the hour. Maybe we'll just close with a couple of additional questions. Someone asked, what's a good age to start the drug for it to have the best protective effect? I know that we're very used to the message, start early, preserve the muscle you have because you can't get it back. How early could you start treating with this?
We have treated boys as young as 4. And we have plans for trials in boys younger than that as well. There's certain preclinical studies that we need to do to enable that, and those are done.
Oh, wow, that's great.
Yeah.
So we should expect that in the future.
In the future, not in the near future, but in the future, yes.
Okay.
and because these diseases are marked by loss from wherever you start, right? When you start very young, of course, you've got a lot more function to lose. It's not like if you start later, you don't have function still left to lose, right? So there's never a wrong time, I don't think, to start on the therapy.
Okay. We had someone ask if you can comment on where you are in enrollment for Grand Canyon. Can you tell us how many
Generally,
patients in that trial, how many still need?
Yes, I can't tell you the exact number. Enrollment is going well. We are enrolling in the US. Those sites are essentially all of the US sites are open. It takes us a bit longer in Europe to open sites and the rest of the world. And so we're working on that as well, because it is a global study and we want to enroll across. So we're aiming to complete enrollment around the end of the year.
And maybe we can end with just one last question about how you're thinking about combination therapies, given that we think it's going to take a cocktail approach to best protect and preserve these muscles. How is Edgewise thinking about that?
We think this can be part of that cocktail and actually would add to it, not just in Becker, but in Duchenne. For example, we've started a trial on top of gene therapy, the FOX study, which has been well enrolled, because none of these therapies are curing the disease, basically. So we need more, clearly. I think all of the experts believe that. And this is a distinct mechanism of action, and it is addressing this contraction-induced injury, which is still there if you're on steroids. It's still there if you have had gene therapy. Somebody asked about givinostat, and we are looking into that. I think that people don't have, at this point, a lot of familiarity with that, and we will be looking at that in the future and how to incorporate that, because all of these therapies, I think, are going to be in a very good place for the community. We're including people on exon skipping therapy in Duchenne as well. We've already been doing that. So it's a very different landscape.
Well, I just want to thank you, Joanne, Alan, Sarah, the Edgewise team for, first of all the work, pioneering work you're doing in Becker and Duchenne muscular dystrophies. We are thrilled to have you with us today. And obviously you can see the community was very engaged, has a lot of questions, is very interested in the work that you're doing. So thank you so much for spending the past hour with us. We will post this recording for the rest of the community and share it broadly for the Becker community. And on behalf of PPMD, I just want to thank you all for joining us today. Thank you.
For all you do.
Thanks a lot. Really appreciate the help.